Hi everyone, my name is Katie Kayan.
And I'm Sarah Ramirez.
And I'm Anna Hashimoto.
And so the research question that we will be answering with our proposal is, how can the study of the degeneration of neurons in Alzheimer's disease be used to stop neuronal damage?
So, the degeneration of neurons can be studied using cryo-electron microscopy and fluorescence microscopy. Our technique can be practiced in labs and tested and of use to scientists. The how and why neuronal damage is still somewhat unknown, and therefore, sadly, there's no cure for Alzheimer's. The disease is troublesome for patients and families all around the world. Therefore, we search for answers to progress the Brain Initiative and discover more about neuronal damage. So what is Alzheimer's disease? It is a condition that impairs memory, thinking, and behavior, and it's caused by the buildup of proteins called beta amyloids, and they form plaque in the brain, making it difficult to think clearly and remember things. There's also another protein called the tau protein that causes tangling and also messes with thinking processes. This leads to degenerative neurons, and therefore the brain is shrinking because it's made of neurons. Yeah. Alzheimer's is a type of dementia. There are 3 broad stages of Alzheimer's. There's preclinical, which would be before many symptoms like forgetting, and there's mild cognitive impairment where the Alzheimer's develops, and then you start experiencing symptoms, and ultimately dementia. And like I said before, there's no cure.
So just to give you guys a little background about what prions are a type of microbe, and they stand for proteinaceous infectious particles. And like its name suggests, prions are proteins. However, there are 2 forms. One is the misfolded form, and the other one is the normal kind. And the misfolded kind can cause the normal prion proteins to develop into misfolded ones by causing a deformation in a region known as alpha helix 1. And so the prion that is specifically involved in Alzheimer's disease, which is what we're looking at, is called beta amyloid protein. And this beta amyloid protein accumulates in the brain, forming plaques, like Sarah said.
So as of last year, nearly 44 million people around the world had Alzheimer's. And it's common. There was also a study that said 30% of people with Alzheimer's also have heart disease, and 29 also have diabetes. So we know that Alzheimer's causes the degeneration of neurons, and therefore scientists using our techniques, we're trying to stop the progression of neuronal damage before it occurs and spreads, to inhibit further damage that can worsen someone's condition. Yeah.
Okay, so I'm going to be talking about some of the studies that have been done by scientists and things that they found to try to advance the cure for Alzheimer's. So one of these is a study that was published in Nature magazine just last year, and it was talking about how scientists were able to discover the structure of the tau protein. And the tau protein is one of the proteins in the brain that is one of the major causes of Alzheimer's because it tangles inside the brain. And so they were able to study the structure of the tau protein using cryo-electron microscopy, and they're hoping to develop new drugs that could perhaps target this tau protein and destroy it. However, one of the setbacks to this approach was the fact that the tau protein is involved in creating microtubules, which transport materials within the brain, and as a result, destroying these tau proteins could have some unintended consequences. Another one of the studies that we looked at was the discovery of the role of exosomes in spreading Alzheimer's disease. So exosomes, they basically transmit information inside the brain, and they contain genes and proteins that can be absorbed by other cells. And so in one of the studies, the scientists purified the exosomes, which still contain the beta amyloid protein, and they found out that these exosomes could still be absorbed by normal healthy cells, meaning that the Alzheimer's disease, the beta amyloid protein, had spread to other cells. Another one of the studies was the connection between viruses and Alzheimer's. So there have been research has shown that the level of the HIV virus in people with Alzheimer's is twice as high as in normal people. And however, scientists don't know for sure whether this link between HIV and Alzheimer's is correct, but there is interest in creating antivirals as a potential cure. And the last study that we looked at was the depletion of an enzyme called BACE1. this enzyme produces the beta amyloid protein that is one of the major causes of Alzheimer's. And so scientists reversed the formation of this enzyme, and as a result, were able to halt the progression of Alzheimer's. And however, this approach has only been tested in mice, so scientists are unsure if it will work in humans as well. So for our project, we plan to study the degeneration of neurons in the brain in order to help researchers better understand the degeneration of neurons and perhaps create a cure that would stop neuronal damage. So we plan to do this using two types of microscopy techniques. One is cryo-electron microscopy, and the other one is fluorescence microscopy. For cryo-electron microscopy, we're going to view slices of brain from mice in order to better understand how neuronal damage affects the brain as a whole. And the second follow-up technique to this would be fluorescence microscopy, and this would be our main approach, which would to be able to study the activity of the neurons and monitor their progression. So we plan to insert exosomes into the brains of mice in order to give them Alzheimer's so that we'll be able to monitor the progression of the disease from the very beginning to the end. And we plan to create a fluorescent dye that would target calcium because when neurons fire, they release calcium. And in order to monitor this neuronal activity, we plan to use this fluorescent dye to be able to see when the neurons slowly stop firing and die off. And secondly, we plan to be able to design another fluorophore, which would target the prion proteins inside the brain, or the PRPSC, which has membrane glycoproteins that would allow the fluorophore to attach to it. And in doing so, scientists would be able to better view the prion protein and understand it.
Okay, so we have proposed to use cryo-EM to observe the degeneration of neurons in mice brain samples. However, because cryo-EM involves freezing the samples before viewing it, we will need to obtain multiple samples of mice brain slices from all stages of Alzheimer's, and that is both very inconvenient and time troublesome. Another challenge we face is that when we monitor the full effect of Alzheimer's through fluorescence microscopy, we will need to begin from the first stages of Alzheimer's to the very last stage. And this process is very time-consuming, and it's a lot of data to process. So to summarize, we propose to utilize cryo-EM to view the brain slice samples of mice affected with Alzheimer's. And through this data, we will be able to understand how the degeneration of neurons affect the brain at different stages of Alzheimer's. We will also plan to create a fluorophore that would attach to the calcium 2+ emitted by the neurons in the brain. Through fluorescence microscopy, we will be able to view the fluorophores, which the amount of fluorophores, which actually correlates to the activity of the neurons, and therefore we can observe the degeneration of the neurons through the changes in the amount of fluorophore. We will also create another fluorophore that will target the PRPSC, or also known as the cellular prion protein, and bind it to bind to it to affect the neurons with Alzheimer's disease and make it fluoresce. And the combinations of the two techniques will enable us to study the structure and monitor the degeneration of the neurons. And through the two techniques, we hope to collect information on the activity of the neurons and how they deteriorate through Alzheimer's. And we hope to provide researchers through our data, we hope to provide researchers with information on how they may be able to find the cure for Alzheimer's in the early stages of Alzheimer's.
So here are our contributions and our references. So thank you. If you have any questions.
Yes.
So we chose to study mice because mice have been used in other research to study Alzheimer's, and also because mice often resemble humans, and also because of the ethical considerations of using larger animals. For example, mice, they're smaller and easier to access and more convenient to use. And also by putting Alzheimer's or genetically engineering the mice to have Alzheimer's, they have been shown to show the same symptoms as humans would have if they had Alzheimer's disease.
Any more questions? Yes.
So to go off on the previous question, do you think it will be safe to infer the data from the tests of the mice to actually use that data to treat humans? And maybe because they're completely different. Well, not completely, but they are different things, and you can't really just use data from a mice to treat a human, and because you can't really test a human, you can't really give a human Alzheimer's. So, do you think it will be safe to use the data?
Okay.
So I think when the ultimate goal is to eventually test it on humans, so I'd say mice first, and then once we gather enough information and we're sure how to apply it or convert it onto a way that's safe for humans, of course it'll be tested first. Like some people, they volunteer for these things, or they need like something right away, so they can try dangerous new methods. But ultimately, the goal is to use this method on humans eventually. So we won't do it unless we know it's for the most part safe.
And also, a lot of researchers, they've been studying Alzheimer's through mice. And this is because, like I said earlier, the mice, when they are given Alzheimer's, they show more symptoms that are related to humans if they had Alzheimer's too. They
also have similar structure, like their brain, their organs, so that's why we use mice. Yes?
What do you hope to do with the information that you would get from doing this kind of research?
So currently scientists, they don't understand, or they know that neurons degenerate with Alzheimer's. However, they're trying to find a cure in order to stop this damage from occurring before it starts. So we plan to use our research to be able to help scientists better understand how these neurons degenerate through Alzheimer's and perhaps monitor their progress and activity. Yeah.
So we have one more question from Dr. Ventalillo.
It's a very elegant proposal, and what I really like is that you actually brought in two techniques to try to address the research problem, which is really nice. So if I understand correctly, you said you're going to use brain slices, so kind of a fixed sample, use electron microscopy, and then more live imaging, right, using fluorescence, which makes a lot of sense. And I wanted to ask you, do you think it would be possible to actually use both techniques almost at the same time? Maybe not at the same time, but maybe one after another on the same type of samples, and what would you learn from that?
Yes, I think it would be possible, but for the cryo-electron microscopy, the brain samples, they would have to be frozen. So for the fluorescence microscopy, we plan to test it on live mice, and then perhaps after the disease has progressed and towards the end stages, we could use the brain samples from the mice that had the Alzheimer's and test it under the cryo-electron microscopy to see the effect of how the damaged neurons affected the entire brain.
Right, so it takes the same samples basically and get them first with the fluorescence and then
Yes.
All right. Thank you, ladies.
Thank you.
Thanks.